My name is Travis Cool, and my 2010 Symposium project is about the possibility of finding soft tissue in T-Rex femur. This specific T-Rex femur was found in a sandstone formation in Hell Creek, Montana in 2003 by Jack Horner. It was later examined by scientist Mary Schweitzer, where she believed that she thought she saw what looked to be soft tissue inside of the bone. This is so amazing because soft tissue has never been found before 68 million years ago, which is located in the Cretaceous period right here. The first method that was used on the bone utilizes a machine called a gas chromatography mass spectrometer. What this does is it breaks down the bone into a gas and it sorts out the various makeup of the bone by the gas depending on their mass and their charge. The second test utilized a liquid chromatography mass spectrometer. This is very similar to the gas chromatography mass spectrometer, except instead of breaking the substances down into a gas, it broke the bone down into a liquid. Also, peptides were found. What a peptide is a protein fragment, and scientists developed a database that sequenced the peptides, which is basically just putting the proteins together to make sure that they're intact. The third method uses a microscope called an optical electron microscope. This is different from conventional microscopes because it uses electrons instead of transparency and passing light through an object to magnify an image. Also, antibodies that were raised against ostriches were applied. What an antibody does is it binds itself with foreign protein so that scientists will know if there is, in fact, protein in the bone and if it is intact and present. The last test utilizes the optical electron microscope stated above. Also, it uses what's known as an atomic force microscope. This has a spring-mounted probe with a laser tip at the end of it. This can view individual atoms inside the bone. Also, this test also exposed the bone to antibodies raised against the birds. The results from the first test is that soft, moist bone marrow was found. As you can see in picture A right here, the scientists stretched the bone and it retained its elasticity, so that is how well the bone has been preserved. Also, in this picture, it is kind of hard to see, but these three arrows are pointing to what looks to be red blood cells, although it was not confirmed. These blood cells look very similar to those of a modern-day ostriches. This is the results from the gas chromatography mass spectrometer test. The results from the liquid chromatography mass spectrometer test is that this is a test that tried to assemble the protein fragments, and they were able to sequence 7 different proteins. And as you can see in this graph is made by a liquid chromatography mass spectrometer. What this does is it makes a graph, and for every line is a various chemical, and if the higher the line is on the graph, the more present that chemical is in the substance, in this case the bone. As you can see, glycine, proline, and lysine are all present in this bone. These 3 chemicals make up the triple helix structure in collagen, which is the main protein found in bone. So it's confirmed that protein was found. The results from the optical electron microscope test is that the ostrich antibodies interacted with the T-Rex tissue, showing that the ostrich is probably one of the modern descendants of the T-Rex. Also, 7 peptides were found and were very similar to the ostriches. And what was interesting about this test is that the T-Rex bone took longer to erode in the acid than it was expected. This is showing how well preserved this bone was in the sandstone formation. Results from the final test is that a T-Rex bone matrix was found. What a bone matrix are various cells that contain collagen fibers, inorganic salt, and ground substances. These binded very strongly and frequently with the ostrich antibodies. Also, it did bind with the chicken antibodies that were applied, but it did not bind as well and not as frequent as the chicken antibodies. So scientists believe that the ostrich is a closer relative of the T-Rex. And after this test was done, another scientist had questioned its authenticity, so another test was done to make sure that the results were accurate. results from this other test, or that overall it confirmed the results and that the ostrich is more closely related than the chicken. As you can see, D, H, and J, these three pictures are from an ostrich, which looks extremely similar to the rest of the pictures that are from the T-Rex bone. These three arrows again are pointing to what looks to be red blood cells in the T-Rex and what are confirmed to be red blood cells in the ostrich. This is so amazing because nothing like this has ever happened before, and the paleontologists are trying to figure out, why this happened, because this is the first near living tissue sample from 68 million years ago. And although it was disappointing that no DNA could be found from the T-Rex bone, because if DNA could be found, it is a possibility that a T-Rex could be cloned from a ostrich. These findings don't affect our everyday life, but they do affect paleontologists, because with this new tissue information, the modern descendant can be determined to be the ostrich. And now, with these new tissue samples, much can be learned and examined with these tissues, so that the T-Rex skeletal system can further be researched. The main question that comes to mind is, why is this important? This is so important because these methods have never been used before on a bone like this. So now scientists can go and do these same exact methods on different bones to see if they can find different results, so they can compare and maybe find out more about the specific T-Rex that the tissue was found from. And since no DNA was found, it's likely that a lot of scientists are going to start looking for DNA to see if they can unlock more mysteries about the T-Rex. These are all my references. The main two references that helped me the most were the ones by Mary Schweitzer and John Asara. These are the two scientists leading the operation on this specific femur, so they were very helpful. And now that scientists have found tissue from over 68 million years ago, is it possible for DNA to be found so that a T-Rex could be cloned, so living studies could be conducted on one? Is there any questions?
Probably indicated where this bone was found. Could you tell me kind of where this was found, such that
The T-Rex.
the unusual occurrence occurred?
Yeah, the T-Rex bone was found very deep in a sandstone formation in Hell Creek, Montana, 2003. Jack Horner.
I noticed that you observed that there were different theories that scientists had, and when the initial research showed that it might have been linked to a chicken versus an ostrich. How prevalent did you find your research to kind of point out that sometimes scientists don't always agree on the theories?
There was a lot, because the previous belief thought that alligators or crocodiles were close, and then someone came out and said chicken, and then there's this big argument between chicken and ostrich, but the last test proved that ostrich is more related than the chicken.
When they chose the ostrich antibodies to react with the tissue, did they react with other antibodies as well, and this was the best results came from the
Yes,
ostrich
they
antibodies?
used chicken and crocodile and alligator and many other birds,
Okay.
but
so,
ostrich was really,
that was a
prevalent.
standout. Okay.
So what is it that got you interested in this question, or this topic?
Well, have you ever seen the movie Jurassic Park? It's kind of what it's like. It's like, something's found in a T-Rex bone, not supposed to be there, maybe one could be cloned. So, it's pretty interesting.
Any other questions? Well, thank you very much.